Hello everyone, I am Snigdha Bharadwaj from ITS College of Pharmacy. I welcome you all in this session. In this particular session, we are going to discuss about brief introduction of genetic engineering. So, let us get started. So, basically genetic engineering is the artificial manipulation or alteration of genes. As in one of my lectures, I have also discussed about protein engineering. Here, engineering is referred as manipulation and alteration of particular material, genetic material. In protein, we generally do alteration in amino acids and nucleotide level, whereas in genetic engineering, it is a broad term. So, it is an artificial manipulation and alteration of genes at genetic level. Genetic engineering involves removing a target gene from one organism, right, inserting target gene into DNA of another organism, cut and paste process. So, basically we are doing modification and alteration at genetic level is termed as genetic engineering. So, recombinant DNA technology or is the basic example of these genetic engineering processes. So, in recombinant DNA, these are prototype terminologies or scientific terms you should understand during we talk about genetic engineering. Altered DNA or recombinant DNA recombines after a small section of DNA inserted into it. So, recombination of DNA, we take two different type of strands and cut the specific sites and merge them into one to get the third variety. Okay. From A and B sources, we get hybrid variety of A and B. So, this is basically called recombination DNA. Recombinant DNA, genetically modified organism, which is also referred to as GMOs, is an organism with altered DNA. So, during that genetic engineering process, we get the species we genetically modified basically is termed as GMO, right. So, A type animal, B type animal, in which we using that particular wild type species, if we modified some sort of modification at genetic level, then the species we are getting from that modification is known as genetically modified organism. So, genetic engineering breaks the species barrier. Yes, genetic engineering allows DNA from different species to be joined together. Of course, we are modifying or altering the genetic level. So, basically allows DNA from different species to be joined together. Why we are doing these genetic modification and alteration? To get the higher functional species, right. This often results in combination of DNA that would never be possible in nature. So, it is not natural. Basically, this process is artificial. We are producing and we are generating the highly active genetic species from the natural sources. So, that is why it is called genetically modified organism or species, right. So, this often result in combination of DNA from two different sources to make the new one, right. For this reason, genetic engineering is not a natural process. If DNA is transferred from one species to another, the organism that received the DNA is said to be transgenic. Yes, from one source we are extracting and isolating DNA and transferring into another one, in which we want gene to be modified and we want different type of functional behavior of that particular organism, then the DNA is said to be transgenic. Examples, you can see in the pictures that the pig has variety, one is of yellow, and this is all because genetic modification. So, unmodified plant, you see it is useful in all majority type of industries, in agriculture, in animals, in therapy treatments, right. So, we can use a human gene inserted into bacterium to get, if human gene is producing some sort of enzyme, particular enzyme. So, if we put that gene and transfer that into a bacterium, then we can expect that organisms can also produce that particular type of enzyme. So, this is all under testing period now. So, some of the experiments have been achieved and some of the experiments did fail due to its procedure complexities. So, it is not that simple. If we talk about modification at genetic level, that means we should have basic understanding about the genes, about its amino acid nucleotides, genetic level, genetic material, genome, cellular, all very thorough knowledge should a person must have to get the desired species. Sometime these experiments can also get failed. A human gene inserted into another animal, yes. A bacterial gene placed in a plant. So, all these type of modification, cross species transfer of genes are these type of examples. We are modifying genes, or we are putting genes from different source to another source, like human to bacteria, plants to humans and all that, right. So, tools. So, in this picture you see this is unmodified plant, the growth of plant is normal, whereas modified has different type of plant, increase efficiency and less energy lost of heat and 15% plant growth. So, all these parameters can be enhanced using this genetic engineering, which is widely used in agriculture industries now. Tools in genetic engineering. So, for achieving a particular process, we must know how can we approach this particular thing. So, first is restriction enzymes. So, restriction enzymes, as I have told you to remember that recombinant DNA technology here. So, it is a key terminology how we use that particular modification using these type of enzymes, restriction enzymes. So, these are special enzymes used to cut the DNA at specific places. Different enzyme cut DNA at specific base sequences known as recognition site. For example, one restriction enzyme will always cut DNA at the base sequence GATTC. So, these are nucleotides basically. So, they have specific nucleotide sequence in particular DNA, and these restriction enzymes always did cut these particular sites which are sensitive to them, or recognize these specific sequence can be recognized by these restriction enzymes. Another restriction enzyme only cut the sequence GATC. So, we can have multiple type of restriction enzymes in that, right. If DNA are from two different organisms is cut with the same restriction enzyme, the cut end from both source will be complementary and can easily stick together, right. So, this means that if we are using two different type of strains and cutting a particular site, so these particular fragments will be complementary to each other and they can be easily stick together. They can be easily to stick together using these particular type of restriction. That means, we can use common restriction enzyme to cut the specific site from two different varieties of the strains, and we can join it together using ligases enzyme, right. So, restriction enzyme is just only to use to cut the specific site we need. Next is DNA ligase. So, here we just cut the site from the required variant, and now how can we join that particular site all together using DNA ligase enzyme, which acts like a glue sticking foreign DNA to DNA of the cloning vector, right. So, it will only work if DNA from the two DNA sources has been cut with the same restriction enzyme. Yes, just now I have told you here, if we are cutting two sites from different variants using same restriction enzymes which have a complementary end, right. So, it can be easily stick together using DNA ligases. So, will only work if DNA from 2 DNA sources has been cut with the same restriction enzyme, that is sticky end of cut DNA will be complementary to each other, right. So, this is how DNA ligases work. Now, process of genetic engineering. What are the different steps involved in genetic engineering? So, 5 steps, basically broad 5 class of processes are there. Isolation of that particular DNA, right, from the organism, right. So, for example, we need one variant and we have the variant. So, we can isolate that particular gene from the organism, right. So, this is basically gene or DNA, right, we have extracted or isolated. Now, cutting of the specific site. Now, insertion or ligation, yes, if it is 2, then second one joining them together and transformation into one, and then expression. That means different function behavior. So, this is how we attain the process of genetic engineering. First, isolate the DNA which needs to be rectified, or gene, cutting the required site or sequence, insert it into the other species using ligases enzyme, transformation, and then expression will get changed. If the genetic sequence will get changed, automatically the phenotype of the species will get changed. So, note the following example will explain how a human gene is inserted into a bacterium, so that bacterium can produce human insulin. As I have told you, if some enzyme is producing by human cells, if we genetically modify using this genetic engineering process, if we transfer this process to a bacterium, then that bacterium can easily produce that insulin. So, here you see process. So, these are DNA of 1 and DNA of second. So, we cut the specific site here, you see TGACC, CTGGA. So, here you join both of the sticky ends. These are sticky ends because these are cut complementary to each other and cutting by the same restriction enzyme, and now from this we can get this recombinant DNA. So, using restriction enzyme or ligases enzymes, so we can have two different type of combination applications. What are the basic applications of GE? Weed killer resistant crop. First of all, in agriculture, in regards to that plant or agriculture industry, we can use these genetic engineering process for weed killer resistance crop, to develop weed killer resistant crop. Those who are affecting by the weed or insects, we can modify it using some genetic engineering processes and to get more resistant crop there. So, many types of crop plant have bacterial genes added to them, right. These genes make the plant resistant to certain weed killers, herbicides. So, if we incorporated bacterial genes into the plant, so we can expect the variety of that particular plant is now resistant to that particular weed. This means that the weed killer kills the weed, but not affect the transgenic plant, because we have transferred bacterial DNA to the plant cells, that is known as transgenic plant now. In animals, there is a growing trend to experiment with inserting human genes into the DNA of other mammals, right. There is a growing trend to experiment with inserting human genes into DNA of other mammals. So, human genes transferred to the mammals, mammals. So, it is also giving some positive results in researches. The transgenic animals formed in this way will then produce a human protein and secrete it into their milk or even into their eggs. So, we can use it in that particular way to produce this particular enzyme through animals. So, we can transfer it into mammals. Example, sheep produces human clotting factor. So, we are getting medicinal benefits using this approach. A human gene has been inserted into DNA of sheep. This allows the adult sheep to produce a clotting chemical needed by hemophiliacs to clot their blood produced in the milk of sheep. So, this is the basic example. If we insert a human gene, a human DNA into the DNA of sheep, then sheep can start producing clotting chemical or clotting factor, which can be utilized by hemophiliacs, which are lacking to clot the blood efficiently. So, we can use for that particular treatment. Yes. So, farming basically is the production of pharmaceuticals by genetically modified animals. So, here what we are doing, by inserting human gene into the animals, we are producing the human factors or medicines. So, this concept is known as farming, PHARMING, right. So, here you see human gene is transferred to the mammals, and then egg, injection of human gene, development within the host cow, transgenic cow. Yes. So, these are egg donor eggs. So, we can use injection of human gene into normal cow, and now we can have this transgenic cow with better functional behavior, right, milk from transgenic cow. So, we can compare the effect or the properties of the milk produced from the normal cow and from the transgenic cow. Microorganism, bacteria make insulin. This allows the bacterium to produce insulin for use by diabetes. So, we have discussed plant, we have discussed animal, now we are talking about microorganism, how this genetic engineering process is useful in microbes. So, bacteria make insulin. This allows the bacterium to produce insulin for treatment of diabetes. So, human cell, here you see human cell, insulin gene you have isolated. Now, this is bacteria, and then you have isolated plasmid, genetic material we can say. So, you have put using restriction enzymes, you cut the required site, specific site, and now you have made recombinant plasmid, and you have inserted this plasmid into transgenic bacteria. Transgenic, why we are saying, because we have inserted a recombinant DNA to that particular species. Now, if we grow that bacteria in culture, so we can expect the human insulin produced by this transgenic bacteria, right. So, this is how we can incorporate this technology at microbial level. Now, what are the different ethical issues? We are in one side we are saying that genetic engineering has positive effect in all area, right, but on the other hand we must have a standard protocol for conducting these kind of genetic engineering processes. But what are the basic hurdles, what are the ethical issues we are facing, so that we can have efficient use of this technique in all type of areas. The release of GMO into the environment. Yes, GMO, genetically modified organism. So, once we transfer a genetic material, or once we alter the genetic material from one to another source, then the releasing of that GMO into the environment is might be at risk, right, because we do not understand the basic difference initially. So, we have to keep it under testing period or something like that, so that we can have full understanding of the basic mechanism involved in the organism, the changes, what are the positive, what are the negatives, so that we can expect its performance, biological performance we need to understand once we produce genetically modified organisms. Because in here we are just focusing on the positive aspects, but during genetic engineering, playing with the genetic DNA material is not a simple thing we are incorporating. So, we are changing whole genome of an organism by just transferring one species strain to the other, right, genetically modified. So, releasing that particular GMO at initial level. So, we need to keep it under investigation for a longer period, so that we can have a basic full understanding about that particular organism we have produced, right. Danger of possibility of GMO being released into the environment. So, yes, of course, there is a possibility that if we release that species into environment before knowing its bad aspects as well. So, we can expect that it might harm the environment, right. GMOs grown in the bioreactors do not provoke as much fear, right. So, bioreactor produced GMOs do not have much effect on the environment. GMO grown outdoor, fears of foreign genes they contain being spread to other plants. Yes, spreading is quite possible here, right. Animal welfare. So, there is a serious concern that animal will suffer as a result of being genetically modified. You can search online, you may have find different genetically modified species with very severe adverse effects after being genetically modified, right. So, these are basic concern why the government has issued with this type of particular process, because we are not sure what we are developing, what we are changing at genetic level will get the desired result or not. Use of growth hormones may cause limb deformation and arthritis as animal grow. Yes, these are some negative effects in the genetically modified organisms. So, ethical issues are there. GMO as a food source, as outlined below, some of the fears associated with the use of GMO as a food source. If we use genetically modified organisms, what will be the bad negative aspects of foods, like cannibalism. Cannibalism, eating an animal containing a human gene, is a form of cannibalism. It is a type of disorder. Feeding GMO containing human genes to animal that would later be eaten by humans. Again, then the genetically modified organisms were made by inserting human gene into that particular animal, and then again it is consumed by human species, and that genetically modified organism is again consumed by human species, that is also inside the humans, that will affect the total physiology of that human specifically. So, there are religious reasons also. Eating cow genes that are inserted into sheep would be offensive to Hindus. So, some of our ethical issues written here, some of not. Offensive to vegetarians or vegans, eating animal genes contained in food plants cause concerns. Yes. So, vegan are considered to be pure vegetarian, and if we transferred plant bacteria or animal organism or genome into the plant sources, so it is considered that it is not purely vegetarian. In genetic engineering in humans, the following issues are a cause for concern. If tests are carried out for genetic diseases, who is entitled to see the results? Yes, if we are doing modification in humans, then who is responsible for knowing all the negative and positive aspects of the diseases, right? So, are the risk considered properly, are the treatment available properly if any negative effects comes out after this genetic engineering. So, these are basically concerned. Insurance, lending companies, will they insist on genetic test before they will lend money to a person? Test on unborn babies, could this lead to abortion if a disease is shown to be present? So, these are different the side effects. Basically, ethical issues are depending on the side effects, which are considered to be a more important aspect of this genetic engineering process. Need for legal controls. There are no standardization procedure as such for human beings to insert human enzyme into the other sources of animals, food and plants, right. Development and expansion of eugenics. So, this is another term in microbiology. So, these are some ethical issues which can be faced by a firm or organization which are involved in these genetic engineering processes, but this is not the end. Yes, we are focusing more on the positive sides of the experiment, and now definitely in coming future, if we develop a standardization procedures for this particular technique, we will be able to deliver the exact and positive aspects of the technique into the various industrial applications. So, these are references. Thank you. I hope you liked my presentation. Stay safe and take care.